ready for the last tutoring ever? I know you guys are sad. We'll see about that. Do you guys have neuro next try? Yeah, you try twos. Oh, you guys try, yeah, so you skip a try. You try twos, right? Okay. Thanks. I don't know. I'm 3 tries ahead of y'all. Yeah. You guys have your 3rd exam today, right? Or not today, this week?
Wednesday.
And you go through fat-soluble vitamins? Sweet. So we're way ahead of schedule, which is what I like to hear. All right, so questions on previous stuff. This is going to be a thoroughly thrilling lecture where I tell you to memorize stuff again. We're going to go through the microminerals. Shouldn't take us too long, and then we'll see what we can do for you all afterwards. I did not bring the mock test. The mock test is on the website. You guys fail at this. You guys Google Drive? Oh, yeah. You can't find the link? I don't know where the link is. It's probably... All right. Hey, hey. All right, what's your email address? Okay. And you're at Parsons? Do you want your...
What?
How about your Parker one?
Oh, at Parsons. I don't know. I just thought you wanted my
Bam.
email.
There you go. You guys now have the link. All you have to do is click on the link, and it takes you to the Biochem folder. And the Biochem folder has Biochem 2, and then I think a mock test section. Check it, so you can stop whining at me for not having the link. It works? And you can see the mock test?
I just saw your folder.
Okay.
I went with it.
Yeah. Mock tests are in there along with all the stuff that I have for the testing series. Cool. And the key's separate, so you guys can take it and check it. I think I did pretty well on these mock tests. So make sure you take those and, you know, study whatever you have to get through this 3rd test. But you're almost there. You're almost done with Biochem. Then you don't have to think about it until try 5.5 when you get to take boards. All right, you guys are excited? Come on. We've got like 1 more hour, I'm hoping, of lecture. We don't have a tremendous amount of stuff to go through. And then we'll talk about whatever you guys want to talk about for a little while. And then I'm going to go home and laminate stuff for neuro. So, all right, so we're talking about trace minerals. We left off on macrominerals. We finished macrominerals last time. So now we're in lecture number 14, page 185. We're talking about the trace minerals. Remember, macrominerals are those minerals that are required in greater than 100 milligram quantities a day. If you need more than 100 milligrams a day, it's a macromineral. If you need less than 100 milligrams a day, it's a micromineral. So if for some reason he asks you a dose on the test, and you I don't know which doses are on this chapter, I don't remember. I have most of them TQ'd, which means he probably told us to know them, unlike the vitamin section, so make sure you're on top of that. Ask him which ones he wants you to know, if he wants you to know them all. But if you know which ones are your macrominerals and which ones are your microminerals, or just memorize one of them and know that the rest must be the other, then you know what the general dosage is. So don't pick something that's less than 100 milligrams a day if you know it's a macromineral. That wouldn't make much sense, right? Cool. Just general good know-how stuff. Same thing as before. You need to know not so much the food sources. You need to know what it does, what reactions it's important in, what enzymes it's important in, how we might get a deficiency of it, and what a deficiency or a toxicity of it might cause. Deficiency or a toxicity of it might cause, the lack thereof or the excess of. So, your microminerals are iron, zinc, copper, selenium, iodine, fluorine, chromium, manganese, cobalt, and molybdenum. Most of those are pretty short. A few of those, like iron, we're going to spend a little bit of time talking about. Most of them aren't going to take us too long. Oh, it was on the screen. Okay, so mineral interactions. Some of them, different things interfere with their absorption. What helps minerals be absorbed? What kind of conditions? Acidic conditions. You need acidic conditions to absorb minerals, period. You need acidic conditions to absorb minerals, period. So, what is a really common thing that people take that's going to lead to the malabsorption of minerals? Antacids, aka Tums. If you take Tums, it decreases the amount of acid in your stomach. It makes your acid reflux feel better. Not so much because you don't have acid reflux, but because it no longer burns. But it decreases your mineral absorption because you lower the acidity of the chyme that's leaving your stomach. And then when we hit it with the bicarb in the pancreas, or I'm sorry, in the small intestine, it drops the acidity so much we can no longer get the minerals out of it. So, you need it to be acidic to absorb minerals. Moving right along to iron. Iron, you need to know the two-letter abbreviations for these guys. I don't think it comes up a lot, but he may casually mention one, not even thinking. You need to understand the two-letter abbreviations, especially for the big boys. Sodium, calcium, potassium, et cetera, et cetera. Iron is Fe. You should know that by now. It's present in every cell in the body in a small quantity. Random fact is that a 70-kilogram adult, which is approximately how much weight? Nice job, 160, about 160, 2.2. Contains about 3 or 4 grams of iron, so not a tremendous amount, but a bit. It's needed in every living cell in the body. Maybe a TQ, maybe not so much. And its major use, definitely a TQ, is for what? Oxygen transport. How do we transport oxygen? Hemoglobin and myoglobin. Does myoglobin transport oxygen? No, it does what? Stores oxygen. Good. Exactly what I wanted to hear. So, major uses for oxygen transport. It's a major constituent of the heme proteins, hemoglobin, myoglobin, and cytochromes. What's a cytochrome? Part of the electron transport chain, which moves electrons and helps with oxygen. And it's also important for monooxygenase and dioxygenase. Why? Because it binds oxygens. What do you think a monooxygenase and a dioxygenase do? They move oxygens. What moves oxygens? Iron. Hemoglobin on iron. So iron's a big guy that binds oxygen. So anything that you need to bind an oxygen in, you need to have an iron around, or you could have an iron around. It helps a lot. I also wrote vitamin C in here. Why?
Are those the active forms of vitamin C?
No. What's the active form of vitamin C?
Ascorbic acid.
Ascorbic acid. Good. But why did I write vitamin C by that? You need vitamin C to do that because those are some of the reactions that vitamin C does. Vitamin Z does the collagen hydroxylation, monooxygenase, dioxygenase, stuff like that. Those were two of the enzymes that you needed vitamin C for. It's a coenzyme for vitamin C. Vitamin C is a coenzyme for those enzymes, monooxygenase and dioxygenase. It's used in the FES cluster proteins. That's part of the electron transport chain again. FES clusters are in there with the cytochromes. They move the electrons around in oxidative phosphorylation, so don't worry about it too much. It functions as a cofactor in many reactions. This is important. Ribonucleotide reductase and peroxidase. I should hope you can figure out that peroxidase needs iron. That should be an obvious one. What's ribonucleotide reductase? Purine pyrimidine biosynthesis. It makes DNA. It makes the deoxy versions. Remember, this little guy is the guy that took it from UDP, that's a bad example, TDP to DTDP. So it took it to the deoxy version so you can make DNA. And I told you way back when in chapter 9, that ribonucleotide reductase was an iron-requiring enzyme. That was a TQ back then. It's a TQ now. It's just in the other direction. Ribonucleotide reductase is an iron-requiring enzyme. Peroxidase, you need to write this in, and it'll come up later, though, is a.k.a. ceruloplasmin. Peroxidase, just for your reference here and later, is ceruloplasmin. We'll talk about its function later. So, the proteins of iron transport. You guys have probably talked about these before. If not, you're going to hear them now, and you'll hear a lot of them in like lab diagnosis and nutrition and stuff like that. We have three major transporters of iron. One's the storage. We have transferrin. What do you think transferrin does? Transports it. It's the blood transport. We have mobile ferrin, which is the intracellular transport. And then we have ferritin, which is the storage form. So you need to know a couple of different things in here. Transferrin transports iron in the blood. Easy enough to figure out. It's a plasma protein, a.k.a. it's in the blood. And don't worry about that. You guys remember this? Apotransferrin? An apoprotein and its coenzyme make a holoprotein, right? Me apo, you holo me. So, apotransferrin is transferrin without any iron bound to it. It's just the protein part. Remember, apoproteins are just the protein part. They lack the cofactors and the coenzymes. So apotransferrin is transferrin without iron. Please know that. Apotransferrin is transferrin without any iron attached to it. When it gets iron, it goes from apotransferrin to transferrin. Very importantly, all of these guys bind Fe3+. All of these guys bind Fe3+, period. Is Fe3+ ferrous or ferric?
Wait, it is more.
Ferric. Why? How do you remember ferrous? How many people do you want on a Ferris wheel? You want 2 people on a Ferris wheel. You don't want 3 people. So Ferris wheel, ferrous iron, 2+. Ferric iron is 3+. It's the other one. And she's right, ferric iron is always an extra oxidative state above it. So easy way though, Ferric or Ferrous for ferrous. So ferric iron is the bound form. Ferrous iron is the free form. So when these guys pick up 2 Fe3+, they become transferrin. They go from apotransferrin to transferrin. Now please note, he will ask you a question, and this is important too. They bind 2 iron Fe3+. The number 2 is important, because he will ask you a couple of questions on the test, and one will say, how many iron units are bound to an apotransferrin? How many iron units are bound to an apotransferrin? 0. It's apotransferrin. How many iron units are bound to an apotransferrin? How many iron units are bound to a transferrin? Fantastic. How many ferrous iron units are bound to a transferrin? How many ferrous iron units are bound to a transferrin? So make sure you know that they bind the ferric state. Yes, ferrous is Fe2+, and ferric is Fe3+. They bind the Fe3+ version. They do not bind the Fe2+ version. Transferrin. And ferritin and mobile ferrin. They all bind Fe3+. Easy to remember. Just memorize it. So make sure you know whether it's ferrous or ferric, and he usually doesn't ask that, but he could. But you do need to know the numbers, because we're going to talk about ferritin, and ferritin stores a different number, and it's been on the last few tests. It was on when I was there, and I hear about it pretty much every year. Is how many does apoferritin bind? How many does transferrin bind? Good. Or I'm sorry, apotransferrin and transferrin. Mobile ferrin's present in the cytosol. It's the cytosolic absorption pool, not a big TQ. But ferritin, I guarantee, will show up again. Ferritin and transferrin are the 2 big. Ferritin is the storage form of iron. It binds 4,300 ferric irons. 4,300 ferric irons. Apoferritin does not contain any iron. So, on your test, it will say how many iron ions, it could say, how many iron whatevers does ferritin bind? And it'll have 2 Fe2+, 2 Fe3+, 4,300 Fe2+, 4,300 Fe3+. 4,300 FeF plus for ferritin. Fe3+. Yes. Does that make sense to you guys? I'm not confusing you? Okay. Cool. Denaturation of ferritin. What's denaturation? Yep, just proteins breaking down. Causes the loss of apoferritin subunits. As a result, iron particles are freed in the cells. Iron stored anywhere but these guys is called hemosiderosis. Hemosiderosis, I think it says it right here. Or hemosiderin is the deposit of it. Hemosiderosis is the problems associated with it. So hemosiderosis or hemosiderin is a storage form of iron where it's deposited in the tissues. It primarily deposits in macrophages, FYI. That's the first place it deposits. That's the abnormal deposition of iron in tissues. When you have iron overload, or your ferritin's not working, or you're denaturing your ferritin or something like that, it starts to dump off in the tissues. It's not good. It's a heavy metal. You don't want it in your tissues. Yes, hemosiderin is bad. I mean, you could have a small amount and be okay, but when it builds up, it goes from hemosiderosis to hemochromatosis. We'll talk about that later. So hemosiderin is the abnormal deposition of iron in the tissue. Happens usually when you denature ferritin or something like that, or you get something like what's our iron used for? Blood doing what? transporting oxygen. So what happens if you have hemolytic disease? You can't transport oxygen, but what happens to the iron? It's freed, it's just dumped out there. So it's going to deposit abnormally in the tissue. So you might see something like hemosiderosis in hemolytic diseases or something like that. Cool? Just random food for thought. You guys are enthused.
So what's anemia then? How does that affect iron levels being down? Anemia is not as many blood cells, right?
Anemia is, technically it's a decreased hemoglobin or hematocrit. And there are like 30 kinds of anemia. One of those is iron deficiency anemia, and one of those is chronic iron deficiency anemia, which look a little different. So, but yes, if you have iron deficiency anemia, and we'll talk about it, you get a very specific type of anemia. You get hypochromic microcytic anemia, which means anemia that has small cells and it lacks color, hypochromic microcytic. So remember, the majority of a red blood cell is its oxygen binding capacity. So if you can't make hemoglobin, your cell shrinks. It doesn't need as much space. There's not more protein in it. And the iron itself is what, the bound iron, the oxygen-bound iron, is what gives it its color. So if you don't have the oxygen binding capacity, you don't get the color. So you get a small cell or microcytic hypochromic lack of color anemia. That's coming up in the diseases section. And it's just because of what iron does for the cell. Now we talked about this guy before, peroxidase. What's the AKA for peroxidase? Ceruloplasmin. Now, you need to know it's a copper-containing enzyme. It's like 90% copper, but it's an iron-requiring enzyme. So if he asks you that on the test, what two things are important for ceruloplasmin? Iron and copper. It can come up in either one of those. Iron and copper are both microminerals. It's like 90% copper, so just in case he asks, the majority of ceruloplasmin is copper, but it does require iron. Iron is a substrate for ceruloplasmin as well. What do you think peroxidase does? Oxidizes iron. So what is that? Is that ferrous to ferric or ferric to ferrous? Ferrous to ferric. Oxidation is loss of electrons. Yes, or, well, gain of protons. It's a gain of a proton. Oxidation is loss of electrons. So peroxidase, its charge is going up. It's going from ferrous iron to ferric iron. So what does peroxidase help us do? Store it or free it? What's the storage form of iron? Fe3 plus or Fe2 plus? Fe3 plus. So peroxidase is required for iron storage. So peroxidase, aka ceruloplasmin, is required for iron storage. Normally removes iron from the cells because it turns it from ferrous to ferric, from 2 plus to 3 plus, which allows transferrin to bind how many? Transferrin to bind 2, and ferritin to bind 4,300. Good. You guys following along? Okay. Peroxidase is important for the storage of iron. It removes iron from the cells. All that means is it turns it from ferrous to ferric, so it can be stored, because apoferritin and apotransferrin cannot bind Fe2+. They bind Fe3+. So this guy is required to turn them into the form that these guys can pick up. The other one is ferrochelatase. Where have you guys seen that before? What? Puts iron in the middle of the heme group. Fantastic. It's the last enzyme required in heme synthesis. It takes that protoporphyrin ring and adds iron to it. Ferrochelatase is required for heme synthesis. Do you think ferrochelatase is an iron-requiring enzyme? Yeah, fantastic. It's got iron in the name. Good. TQ. Human absorption of iron is approximately 1 milligram per day. 1 milligram per day. Make sure you know that. Supposedly, according to Dr. Sarkar, and he'll probably talk about this, you absorb about 10% of what you eat, 5 to 10%. So how much iron do you need to have in your diet every day? About 10 milligrams a day, because you want to take in about 1 milligram a day. Now, I will tell you this now, vitamin C, and you can write it wherever you want it, vitamin C is incredibly important for iron absorption. What form of iron do we store? Ferric, which is Fe3+, which is the oxidized form of iron. We absorb Fe2+. Only Fe2+ can cross the membrane. What is vitamin C? What is its one of its primary roles? It's a what? It's an antioxidant. What's an antioxidant? It's a reducer. So what does it do? It reduces ferric iron to ferrous iron so that we can absorb it. absorpted, so that we can absorb it. So if you're having a meal that requires iron and you need to get iron into your blood, what does that meal also need to contain?
Orange juice. Hello.
Yeah, vitamin C or orange juice if you just want to like take a tab of iron and chug some orange juice. So yeah, so vitamin C is important for antioxidant capability. Vitamin C is an antioxidant, which means it reduces it from ferric to ferrous. And ferrous, which is about what we're going to talk about, is what we absorb. So you've got this lovely little chart right here. Iron is stored as ferritin or hemosiderin, liver, bone marrow, spleen. Yes, and some people, I don't want to lie to you and say, I think some hemosiderin is normal. It's just excessive amounts of hemosiderin is bad. Excessive amounts means you can't keep it in ferritin, and you go from hemosiderosis to hemochromatosis, which we'll talk about at the end. So some hemosiderin can be normal, I think, but I'm not positive. Ferritin is the normal storage form. Iron reductase. What do you think iron reductase does? Reduces it. Converts ferric iron to ferrous iron. The ferrous form of iron can be transported across the membrane. So iron reductase or vitamin C can reduce this guy, or vitamin C can reduce this guy. Ferric iron is the storage or bound form of iron. Sorry, ferric iron is the storage or bound form of iron. We talked about that. Make sure you guys remember that. And then down here, here's your intestinal wall. We get Fe3 plus being turned into Fe2 plus, ferric to ferrous by iron reductase, which allows it to be absorbed. Fe2 plus is absorbed. It can be bound to mobile ferrin. It can be bound to ferritin. It can be bound to transferrin. But notice they're all Fe3 plus. And that, I'm not sure if vitamin C does its action on its own, like if it just randomly reduces it, or if it regenerates that enzyme. So that enzyme, I know, takes iron from Fe3 plus to Fe2 plus. I don't know if the action of vitamin C is to do that to iron on its own, or to regenerate that enzyme so that it can do it again. Either way, vitamin C is responsible for the conversion of Fe3 plus to Fe2 plus. And it's doing the same thing, but I don't know if it's doing it as a coenzyme to that enzyme, or if it's doing it on its own. It is an antioxidant. In theory, it could do it on its own, but it might do it through coenzyme action. Okay, some iron deficiency conditions. Anemia. Iron deficiency anemia is, well, I'm not going to say common. It's not the most common anemia. So if you have a friend that's anemic, don't tell them to take iron, because taking iron can actually cause problems, because it's a heavy metal. It's not usually iron deficiency anemia, especially in today's society. There's a million other things that cause different types of anemia. So if you have a friend that's anemic, don't jump on the iron train. Get a blood test, or have them eat more green leafy vegetables and see if it goes away first. Supplement the B vitamins. Remember your B vitamins that caused anemia? What were they? Oh. Yeah, you got a megaloblastic anemia. Two B vitamins cause megaloblastic anemia. B9, B12, megaloblastic anemia, B9, B12. What about microcytic anemia? B6. So, 4 things you definitely need to know that cause anemia for this test in the final. B9, B12, megaloblastic anemia. Yes, or macrocytic, either way. The megaloblastic is a specific type of macrocytic anemia. But B9, B12, macrocytic or megaloblastic anemia, large. B6 and iron, microcytic anemia. So, on here, anemia. Reduced hemoglobin content, which gives it its color, makes them hypochromic. And reduced size of red blood cells because it doesn't have any protein in it. It doesn't have any completed hemoglobin in it, so it's smaller. So with iron deficiency anemia, you get microcytic hypochromic anemia. Make sure you know that. I almost guarantee that'll show up. Microcytic hypochromic. That's a big point for this class and many ones to follow. Microcytic hypochromic anemia. Makes sense, right? You don't have the stuff that goes in the cell, it doesn't have any color, and it doesn't have any size. Some other random stuff. Vitamin A deficiency can adversely affect hemoglobin synthesis. Vitamin C deficiency impairs the release of stored iron. Why? And absorption. What? Impairs. A vitamin C deficiency impairs the release of stored iron. Because vitamin C is an antioxidant, which means it reduces it. And to get iron to be stored, we need to oxidize it. To release stored iron, we need to reduce it. So remember, we use freely and absorb and move Fe2+. But the stuff that's bound, transferrin ferritin, mobile ferritin, is Fe3+. So to get it into storage, we have to oxidize it. To get it out of storage, we have to reduce it. And vitamin C helps with that. Helps with the release of stored iron and the absorption of iron by keeping it reduced. Copper deficiency may also cause iron deficiency because of ceruloplasmin. What does ceruloplasmin do? Actually, first of all, what's the AKA for ceruloplasmin? Peroxidase. So what does ceruloplasmin do? It oxidizes iron. As long as you know the AKA, you know what it does. It oxidizes iron, which allows it to be stored or moved, or stored or used. Stored. Oxidized iron is the storage form. So, if you have a copper deficiency, it causes a deficiency of ceruloplasmin because that enzyme is 90% copper. It does not allow you to store iron, which means you can't keep it around. You don't have sufficient storage to deal with it. You don't have enough when you need it. Questions on that stuff yet? Okay. Ceruloplasmin deficiency can lead to an abnormal iron deposition in cells. So, what is abnormal iron deposition in cells? Or what's iron deposition in cells? What's it called? Hemosiderin. So, because iron deposits in cells normally, and because it's a heavy metal, it's going to have some impact. So the areas that it loves to deposit in are the pancreas, which can have an impact and exacerbate or cause diabetes. Why? It's breaking the pancreas. Why is the pancreas important in diabetes? That's where insulin and glucagon come from. Good. So it lands in the liver. It changes iron metabolism and digestive system. It lands in the retina. It changes vision. It lands in the brain, and it leads to basal ganglia disorders causing Parkinson's. So ceruloplasmin deficiency specifically lands in the basal ganglia, which you'll talk about in neuro, and it can cause Parkinsonian disorders. Cool?
But does it, so it's pretty much the only reason why it doesn't do anything to the pancreas is just absorbing much iron and it slows the pressure and everything else?
Breaks the pancreas. It's heavy metal toxicity. It's a heavy metal. So if you have a ceruloplasmin deficiency, you can't store it in ferritin and all your normal storage sites because you can't oxidize it, which means it dumps off in lots of other tissues, and that's where it builds up and causes problems. Hemochromatosis is on the next page. So pancreas, diabetes, liver, whatever, retina and vision and brain and Parkinson's are big. Make sure you know pancreas and diabetes, retina and vision and brain and basal ganglia are Parkinson's. Those are ones he may ask you about. They're very common boards questions dealing with iron overload symptoms. Aceruloplasmia is a lack of what? Aceruloplasmia. A lack of ceruloplasmin in the blood specifically. Autosomal recessive disease means you don't have ceruloplasmin. You get all the same stuff we just talked about. Because this was a ceruloplasmin deficiency, this is a genetic ceruloplasmin deficiency. So this could be due to copper, this could be due to, or this is due to genetics. Either of them are a lack of ceruloplasmin. Both of them cause the same signs and symptoms. Iron deficiency symptoms, abnormal paleness of the skin. Why? Lack of color. What gives skin its pink color? Blood, which is why when you get cold or when you get nervous or when you squeeze your skin, it goes pale because you don't have any blood running through it. So if you have no color in your blood, you have no color in your skin. Why fatigue? No oxygen. Why tachycardia? No oxygen. Your heart's working harder to get it out there. Did you see it before I put my finger over it? And in the large spleen to try and make more red blood cells. All right. Toxicity of iron, especially for children, can be serious, especially for children. Toxicity of iron is called hemochromatosis. Why chrome? Color. What's iron give you your skin? Color. What's excessive iron give your skin and your organs? Excessive color. So hemochromatosis is iron overload syndrome. in which hemosiderin deposits, remember those hemosiderin, which is a deposit in tissues, are seen in a microscope. They're seen everywhere. It's a recessive genetic disease causing from the overabsorption of iron, 2 to 3 milligrams a day. What's the normal absorption of iron? 1 milligram a day. Visibly seen in the liver, the pancreas, the skin, the joints, and you will see it in the brain. In what part of the brain? The basal ganglia, causing Parkinson's. Good. So hemochromatosis is iron overload. It's the really abnormal deposition of iron. Too much iron, can't deal with it, goes everywhere. Hemosiderosis, total body irons are increased and hemosiderin deposits are widespread. So really, the tier is, you have ferritin is your normal storage. Hemosiderin is the next step of storage. It gets stored in the cells. Hemosiderosis is widespread hemosiderin. And then the next step up is hemochromatosis, which is really widespread hemosiderin. Cool? So ferritin, hemosiderin, hemosiderosis, hemochromatosis. Hemochromatosis is the worst, to my understanding. Yes. Yes. Ferritin. Yeah, anyone want to try? Ferritin. Hemosiderin. Hemosiderosis, which is the widespread deposition of hemosiderin. And then hemochromatosis, which is a really widespread deposition of iron. So ferritin, hemosiderin, hemosiderosis, hemochromatosis. Iron's present in lots of stuff: clams, oysters, and sardines, liver, meat, raisins, prunes, walnuts, spinach, lima beans, and lots of iron supplements. He told us to know most of the RDAs from this chapter: 12 to 15 milligrams a day for women, 8 to 10 for men. That's weird, right? Normally women don't need more than men. Why? So women bleed more than men do. So, yes. Survive continually bleeding. So women need more iron than men because they're constantly regenerating their red blood cells, so they got to keep their iron up. Now, this is a cool point. It's really, really, really hard to iron overload on normal foods, on especially fruits and vegetables. It's really easy to overload on iron if you take an iron supplement. So someone that has moderately high iron can eat all the green leafy vegetables they want, which have tons of iron in them, because your body will not absorb the iron present in green leafy vegetables unless it needs it. But supplements are designed to shove the iron into your cells because you're supplementing iron. So people should be careful about what supplements they take, whereas they can eat as much spinach as they want and just try and increase their iron normally. So, cool fact for you. All right, iron took a while. Let's fly through some of these other ones. They're not nearly as long as iron. Zinc, important in many metalloenzymes. He's probably not going to ask you the specific enzymes, but I highlighted some random ones. It's important for metalloenzymes. Easy enough? Anything metallic. The ones you do want to know: Superoxide Dismutase, or Superoxide Dismutase, is a copper and zinc requiring enzyme. Where else do you think we're going to see this enzyme? In the copper section. So make sure you highlight it and TQ it now. Angiotensin converting enzyme. You guys talked about that in physio yet? Oh yeah, what's it important for? Regulation of blood pressure. Regulation of blood pressure. It causes vasoconstriction. Good. And delta-ala dehydratase. We did talk about that. Chapter 9, heme synthesis. Good eyes. Delta-ala dehydratase. So zinc is important for ACE, angiotensin converting enzyme, delta-ala dehydratase, and superoxide dismutase. What's superoxide dismutase? I've talked about it. I haven't really taught it to you. It's something you might have learned in micro. Remember, it's when the neutrophils make the free radicals to blast the bacteria. It's when they can't handle it, so they break out the free radical gun, and that's the enzyme that deals with that, superoxide dismutase. Cool, he won't ask you it. Intestinal cells absorb it. It's bound to macroglobulins and albumin, probably not a big deal. Zinc absorption competes with calcium, copper, and iron, TQ. Remember I told you, know what it competes with, know what it helps, know what it competes with. What helps iron be absorbed? Vitamin C. What hurts iron being absorbed? Calcium, copper, and? Good. Oh, I'm sorry, zinc, I should say. Zinc competes with calcium, copper, and iron. Don't associate the other ones. So what hurts zinc being absorbed? Calcium, copper, iron. So, therefore, what hurts iron? Zinc absorption hurts iron. Deficiency of zinc, the ones you got to know: poor wound healing, hypogonadism, and a reduced sense of smell and taste. Those are the big three that tend to come up in different places. Hypogonadism, poor wound healing, that's a big one, poor wound healing, and reduced sense of smell and taste. I think it's more a reduced sense of smell, which decreases your sense of taste, because smell is intimately connected with taste. But hypogonadism comes up occasionally too. Cool. That's it for zinc. It's a big one. I don't even have that RDAC keyed, so I don't know if you want to know it. Again, you're responsible for asking about those. Copper, what do you think we're going to see under copper? Two things you got to tell me. Okay, that's true. Two more things you got to tell me. Zinc messes with its absorption. Superoxide dismutase and the one that's 90%. Ceruloplasmin, aka peroxidase, because that's what it does. It oxidizes iron. So, copper is important for oxidase enzymes. The cytochrome system looks a lot like iron. Superoxide dismutase, TQ. He might ask about the cytochromes as well. Cytochromes are important for copper and iron. It increases Fe absorption. Copper increases Fe absorption. I don't know why. I was thinking it had something to do with peroxidase, but it must. But my only guess would be that if you have ceruloplasmin operational in the cells and it's taking iron out of the cell and storing it, the cell can absorb more iron. So, hey, no. Ignore it. Increases Fe absorption. TQ, memorize it, because that's what you want to know. It's important for the electron transport chain, just like iron is, and it's in ceruloplasmin, the copper-dependent peroxidase enzyme. Copper is incorporated into ceruloplasmin, which oxidizes Fe2+ to Fe3+. You guys should know that by now. Deficiency of copper due to excess zinc intake, because they compete. So a deficiency of copper can come from excess zinc intake. And a deficiency of copper, not really a deficiency. There's a problem called Wilson's disease, where you can't make ceruloplasmin. You can't make ceruloplasmin. So you're still absorbing copper, but it's not getting incorporated to ceruloplasmin, which is what uses the copper. So the copper gets deposited in tissues. It actually causes some really cool stuff. You might have talked about this in physio. If not, you will. It causes... Oh, it's not even on here. Oh, we'll talk about it. Okay. So autosomal recessive inherited defect in the incorporation of copper into ceruloplasmin. You cannot put copper in ceruloplasmin. You cannot make ceruloplasmin. You cannot oxidize iron, so that's a big problem too. But it also causes the deposition of copper in tissues, as well as iron, because ceruloplasmin defect causes iron deposition in tissues. But a lack of the creation of ceruloplasmin means you can't use ceruloplasmin to move and transport copper, so it gets deposited in tissues. Affects the liver, the brain, the kidneys, and the cornea. You see some really cool stuff in the cornea. You should look it up sometime. It's called a Kays-Fleischer ring, or Kays-Fleischer ring, K-A-Y-E-S-F-L-E-I-S-C-H-E-R, or something like that. You get a copper, like a goldish ring around the colored part of your eye. So it's not really good to see that. It means you have copper deposition in your tissues. But if you look at the eyes, they have like a golden ring in the colored part of their eye. And that's copper being deposited in the cornea. It also deposits in the basal ganglia and causes lenticular degeneration, which is why it says brain. Basically, metals are bad. You have Kays-Fleischer rings? Heck yeah.
I have a yellow ring in my eye. I never know what it is.
No, I don't think it's that.
Okay, good.
Yeah, I think you're fine. I think you would show some other signs and symptoms. See if I can find it. Yeah, I'll see if I can find a picture. Oh, it's Kaiser-Fleischer ring. Don't take my word for that. I'm not an expert on this. Oh, come on. Yeah, you're going to have to look them up on your own. This thing has no color depth. Kaiser-Fleischer rings. Kayserfl something, and it'll pop up. So you get a cool gold ring in your eye, which looks really cool, but probably not good for your health. Deposits in the cornea and the brain. Brain is important, but cornea is the one that tends to come up on test questions because that's something you'll talk about in physio and path and stuff like that. And you guys are all thoroughly distracted with eyes. No worries. So that is Wilson's disease. Can't make celluloplasmin, can't deal with copper. Can produce dementia and liver failure and lots of other stuff. Menkes disease, not as asked as frequently as Wilson's disease, Menkes disease, is an X-linked disorder of intestinal copper absorption. So, what is Menkes disease going to cause? Toxicity or deficiency? Intestinal malabsorption. You can't absorb copper, so is that a toxicity or deficiency? It's a deficiency. You can't absorb iron, so you don't get iron, so you're in a deficient iron state. No, because it never makes it into your body. Your GI tract is outside your body. It just goes straight through you. You get gold poop. You don't get gold poop. What about Wilson's disease? Is Wilson's disease a toxicity or a deficiency of copper? Wilson's disease is a toxicity of copper because you can't deal with it. It's building up in your tissues. So Wilson's disease causes iron or copper toxic symptoms. Menkes disease causes copper deficient symptoms, such as mental retardation, temperature instability, and infection. Again, not a big deal. Menkes disease is asked less, but it's a deficiency of copper. Yeah, not a big deal. Not a big deal. You guys took that in totally the wrong context. Jesus. So it is a big deal, but he doesn't ask about it as much, which is really, let's be real, the only big deal that you guys care about right now. You don't care about little retarded kids running around with copper deficiencies because they have Menkes. You care about what is Dr. Sarkar going to ask me on the test. Isn't that true? I mean, you have a little bit of care in your head for those little copper deficient special kids. I'm going to get fired as a tutor. It's a good thing this is my last tutoring session. Okay.
This is on our test, right?
No, on your final. Oh, burned!
Not really, but like
This is the last. That's fine.
Listen to 6 hours of you, like again. So, but I'm grateful.
Oh, wait. You had to listen to me? You had to listen to me?
I have to.
That's fine. It's vodcasted, which is a good thing. So, but yeah, this is on your final only. Same with last week's like.
I don't really mean to be disrespectful.
No, that's fine. I don't care. I'm just giving you crap. I don't care at all. If you got to be somewhere else, you got to be somewhere else.
I gotta be at home listening to you. That's fine.
That's cool. That's cool. I thought you said you had to listen to me. I was like, you certainly don't. You don't have to push play.
No, yes, I do.
This and the last one, lectures 13 and 14, macrominerals and microminerals, should not be on your test. It just goes up through fat-soluble vitamins. Up through fat-soluble vitamins. No worries. I'm just giving you crap. I don't care at all, really. You're not going to offend me. Okay, manganese. Manganese is present in the mitochondria. Might be important. That would be one of the few TQs he could probably ask you about this. Manganese is present in the mitochondria. It's important for hydroxylase, decarboxylase, transferase, and superoxide dismutase. What else is important for superoxide dismutase? Copper and zinc. Copper, zinc, manganese. Bam. That'd be a good rule of threes, huh? Copper, zinc. I don't think he'll ask that, but it might be a good one. The one he loves to ask about is manganese is important for the synthesis of oligosaccharides. Manganese is important for the synthesis of an oligosaccharide. Anyone randomly remember what an oligosaccharide is? 3 to 6 monosaccharide units. More than 7 or more is polysaccharide. So an oligosaccharide is a 3 to 6 polysaccharide. Don't care about that. That's not a TQ for this test. But manganese being important for the synthesis of oligosaccharides is very important for this test. That is the big TQ from manganese. Manganese absorption is increased in iron deficiency and can be inhibited by iron. What does that mean? They compete with each other. These competes are going to get to you guys, so make a little list. Iron competes with this. Manganese competes with this. Put that together somewhere. Cool? Manganese deficiency. 2 things. Impaired glucose tolerance and reduced synthesis of oligosaccharides, glycoproteins, and proteoglycans. I talked about the oligosaccharides. The other ones are right there too. That's your big TQ for manganese. Now be careful. It says impaired glucose tolerance, and he can ask you about manganese affecting glucose tolerance. But manganese is not the glucose tolerance factor. We will talk about chromium. Chromium is the primary guy you need for glucose tolerance. But manganese is involved in glucose tolerance, so don't get tripped up by that, cool? I'm warning you now. Manganese is involved. Primarily, though, he wants you to know oligosaccharides, glycoproteins, and proteoglycans. But it's not the primary guy for glucose tolerance. RDA, 1.5 to 2 grams a day. I have that TQ'd. I don't remember which ones he said to know, like I said. Last time I'm going to say that. For real. Selenium. TQ, TQ, TQ. Selenium, glutathione peroxidase. Selenium, glutathione peroxidase. Got to know that. This enzyme has a crucial antioxidant role. What are your antioxidants? A, C, E, S. ACEs are your antioxidants. A, vitamin C, vitamin E, and S is selenium. This is your fourth antioxidant, A, C, E, and S. It's really S, E, E because it's selenium. But selenium is involved in glutathione peroxidase, and glutathione peroxidase is a very, very, very important antioxidant. It's one of the main parts of your detox system. If you don't have glutathione, your liver dies really quickly because glutathione keeps your liver alive. Lord knows you need your liver. Glutathione peroxidase protects against lipid peroxidase-induced damage. It helps antioxidize. So selenium is important for glutathione peroxidase, which is an antioxidant. We've talked about that again. And deiodinase. That's one of the other things you got to know about selenium. Deiodinase is important for the deiodination of T4 to T3. Remember, T4 and T3 are what? Thyroid hormones. And how many iodines does T4 have? 4. And how many does T3 have? 3. Fantastic. So if you need to deiodinate T4 to T3, you need selenium, which is you need a deiodinase, which needs selenium. Good job. You need a deiodinase, which needs selenium. So, synergistic action with vitamin E. Why? Synergistic action with vitamin E. Both antioxidants and very specifically protects against lipid peroxidation. What type of vitamin is vitamin E? Fat soluble, water soluble? Fat soluble. So what does vitamin E protect against? Lipid peroxidation. They both work on the fat soluble guys. They both prevent lipid peroxidation, which is really important in the prevention of atherosclerosis. Lipid peroxidation, the peroxidation of cholesterols that bind to your lumen walls, is the main cause of atherosclerosis. These guys reverse and prevent that. So these guys are really important for cardiovascular health and lipid peroxidation health. Peroxidized lipids are nasty. Like they do a bunch of crap. So synergistic action with vitamin E because it's an antioxidant for lipids. It exists in food bound to amino acids, especially cysteine and methionine. Remember selenocysteine, the 21st amino acid we were talking about? I'm betting there's also a selenomethionine because these guys love to go where sulfur would go. So any sulfur-containing amino acid can just as easily pick up a selenium instead of a sulfur. So selenocysteine, selenomethionine, so we get it from those foods. Okay, Keshan disease. Deficiency causes muscle pain, muscle wasting, cardiomyopathy. But the big one you want to know, and this was asked on our test, was Keshan disease. Selenium deficiency causes Keshan disease, which causes congestive cardiomyopathy or heart disease, causes problems. One of my classmates came up with this. Trust me, this wasn't mine. Kesha's disease is from not listening to enough selena. It makes your heart stop. So if you guys know who Kesha and selena are, that'll make a little more sense to you than it did to me. I know who they are. I'm not going to lie. They exist in my world, but I don't know much about them. So Keshan's disease is from not listening to enough selena, aka Kesha's disease. Keshan's disease is from too little selenium, and it makes your heart stop. Cool? Now you know about Keshan's disease and selenium deficiency and cardiomyopathy. You need to what? Nice. Is that a movie or Selena? Okay. Yeah, like I said, don't know much about it. She is a singer, though, right? I knew that. Jeez, come on. Random toxicity exposures do occur in people that work in electronics and plastics and stuff like that, but they're really rare. You can't really get a toxicity of selenium unless it's being oxidized or being vaporized into the air in like an electronics shop. Toxicity is characterized by garlicky breath and hair loss. Sounds like your perfect date, doesn't it? First date, blind date. Man, this guy reeks of garlic and he's balding. Poor little dude. You might not. I mean, come on. Doctors in you. Is he just a hapless soul, or does he have selenium toxicity because he works in an electronics shop? What are you gonna do?
Or is he Paul after PCP?
You know what? Suck it. She called me bald and garlicky. That's what she did. Bald and garlicky. All right. Any questions on selenium? What disease does it cause? Keshin's disease, which does what? Heart failure. Stops your heart. Makes you want to die. Makes you want to what? Makes your heart stop. I thought you said makes you want to dance or something like that. Okay. Iodine function. What's iodine important for? You guys know this one? Thyroid hormones, big thing. Iodine is important for thyroid hormones. TQ, triiodothyronin, is T3. It's the active form. We talked about this once before. Tetraiodothyroxine, aka thyroxine, is the precursor, the inactive form. So T4 is inactive, T3 is active. Thyroxine is T4. Thyronin, or triiodothyronin, is T3. Regulation of growth, differentiation, and development. BMR. Oxygen consumption and thermogenesis. You got to know all of those. I don't remember if it was this or physio. It came up in a which of these is not regulated by thyroid hormones. Thyroid hormones control growth and differentiation of tissues, basal metabolic rate, oxygen consumption, thermogenesis. They run your body's metabolism. So anything that's involved in metabolism is run by thyroid hormones, which need what micromineral? Iodine. Good. So, sorry, micro, yeah, if I didn't say micro. So here's a random chart. I thought we saw this before, maybe in the thyroid chapter, or maybe it was in physio or something. Randomly, iodine is absorbed. Where are thyroid hormones made? In your thyroid? What cells? Follicular cells. Do you guys remember that? Follicular cells? Follicular cells make thyroid hormones. So this is a follicular cell in your thyroid follicular cell. Iodine is absorbed by an iodine pump. It's incorporated into thyroglobulin, which is the binding protein. These aren't big TQs. He doesn't usually ask much about this. Physio will at some point, so I'll talk about it now. Iodine is incorporated into the follicular cell. It's dumped out into that follicular pool in the middle, and it's incorporated into thyroglobulin. Remember, thyroglobulin, what I talked about before, was it's this huge protein with tons of tyrosine residues. So it's tons of tyrosine residues, and iodine is just dumped onto those tyrosine residues. And then thyroglobulin can be broken down in ones and twos to make mits and dits and tits, right? Thyroglobulin is a huge protein with a ton of tyrosines. You dump iodine onto it, and then you chop off the T4s and T3s as you need them, right? Mits, dits, and tits. And tits. Tetras and tris. So, yeah, it's peroxidized on there. It gets broken down into mits, dits, reverse T3, T3, and T4, and then it's dumped out as T3, T4, and reverse T3. So that's all in here. Again, not huge TQs on it, but I'll show it on the screen just in case. Main food sources, iodized salt. Pretty much no, I think iodine is the most common mineral deficiency in the world. Folate's the most common vitamin deficiency in the world. Iodine is the most common mineral deficiency in the world. But it is not an issue in American society because all of our salt is iodized, and French fries. All of our salt is iodized, which means we get plenty of iodine because it is such a deficiency elsewhere in the world. We were talking to one of the neurodocs. He's a neuroimmunologist. He deals with the neurology of the immune system. And he was talking about trans fats. He had this great quote, and it sucks because I love these things. He's like, yeah. He's like, trans fats stay in your system approximately 412 days. He's like, before your body can eliminate them because they're such a foreign substance. He's like, so space out your French fries. And he just like moved topics. I'm like, oh, I love French fries. Yeah. Yeah, you were at that lecture, weren't you? Brilliant guy. Yeah. So, food sources of iodine, iodized salt. You will want to know, you will want to know that food source. That's one of the food sources you got to know. Food source of iodine, iodized salt. You damn well better get that. He might not put iodized, he might just put salt, but you better know that salt contains iodine nowadays. Yeah, and that's a problem too. You're probably not deficient as long as you eat vitamins, or as long as you eat, it's in most vitamins, many vitamins, but as long as you eat vegetables, you're fine. And the problem is most Americans don't eat vegetables, they do eat salt. So they just give them iodine in their salt to supplement. It's like we put B12 in all the bread now, and we put vitamin D and calcium in the milk, because people don't eat vegetables. And where do you get vitamins and minerals? Fruits and vegetables. And people are like, oh yeah, I eat fruits. I eat the little cans of mandarin oranges and the little fruit cups and that fruit puree that has the maraschino cherries in it and stuff like that. Not puree, but you know what I'm talking about, fruit cups. Fruit cocktails, yeah, that's what I'm talking about. Of course you know what that is. Iodine deficiency causes hypothyroidism. Iodine deficiency causes hypothyroidism. Makes sense, right? You don't have thyroid hormones, you got hypothyroidism. What's it going to do to your oxygen consumption? You decrease it. What's it going to do to your BMR? Decrease it. What's it going to do to your metabolism? Decrease it. I said that, BMR, oxygen consumption. What's it going to do to growth and differentiation of your tissues? Decrease it. What's it going to do to your body weight? Increase it. Because your metabolism goes down, your weight goes up. So the picture of a hypothyroid person is overweight, cold, small, or short, fat, and overweight, or short, fat, and overweight. Short, fat, and cold. That's hyperthyroidism is Graves' disease, and that's exophthalmos, which is bulging eyes. Like Steve Buscemi. I swear to God, Steve Buscemi must have Graves' disease. Those eyes are inane. Like, you should never see the eyes with, like, the whites above and below that as big as that guy's got them. Okay. Hypothyroidism, insufficient thyroid hormones, causes two things. Goiters in adults, or myxedema. I don't know if he talks about this in class, but that is true. Hypothyroidism in an adult is called myxedema, M-Y-X edema. Hypothyroidism in a kid, and this is awesome, is called cretinism, the little cretins. Hypothyroidism in a kid is cretinism. Hypothyroidism in an adult is myxedema. But it will cause a goiter, a hypothyroid goiter, because your thyroid is trying to upregulate. It's differentiating to try and pump out more thyroid hormones, but it doesn't have the iodine. So you're going to get a big old goiter, which is that swelling in your neck from your thyroid working over. So goiters in adults, or hypothyroidism in an adult, is myxedema. Hypothyroidism in a kid is cretinism, and it causes goiters, which is an enlargement of the thyroid as it tries to upregulate and work harder. Guess what? Hyperthyroidism also causes goiters. Hyperthyroidism is when your thyroid works too hard, and that can also cause the swelling of the thyroid. So thyroid goiters aren't indicative of hyper or hypo. But hyperthyroidism is excessive thyroid hormones. The picture of that is skinny, always hot, always sweating, uses a bunch of oxygen, high BMR. They can eat whatever they want and never put on a pound, stuff like that. So iodine toxicity causes the toxic goiter. You have too much iodine, it builds up in the thyroid because your thyroid is really, 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 really good at absorbing iodine. Thyroid is really good at absorbing iodine. So, and guess what they do? If you have a toxic goiter, they might inject you with radioactive iodine. Because where's all the iodine going to go? Into your thyroid, which means it's going to kill your thyroid preferentially. So that's one of the things they can do is just shove radioactive iodine in you because it concentrates so much in your thyroid that it won't affect the rest of the tissues nearly as much. RDA 90 to 150 micrograms a day. All right, we got like 4 short ones left. Chromium. Chromium TQ is the glucose tolerance factor. What helps in glucose tolerance? Manganese. Manganese assists. But if he asks you which mineral, which micromineral is the glucose tolerance factor, chromium. Chromium is the glucose tolerance factor, or a constituent of the glucose tolerance factor. It's important for the glucose tolerance factor. It facilitates glucose uptake by cells. Lowers cholesterol and triglyceride levels, lowers blood glucose levels. It's great for diabetics, right? Chromium is great for diabetics. It's got a role in type 2 diabetes, so they actually recommend chromium in diabetics because it helps them regulate their blood glucose. Now, it can be a problem if you're taking insulin, because what's exogenous insulin do? What does it do to blood glucose? Insulin does what to blood glucose? Decreases it. What does chromium do to blood glucose? Decreases it. So what can happen to blood glucose? Go way down. So you got to be careful, but it's an alternative to insulin, not an alternative, but it can be helpful in not really bad cases. Chromium toxicity, TQ, you usually ask this. The chromium 6+ form is toxic. The chromium 3+ form is okay. 6+ bad, 3+ okay. Almost nobody sells the supplement of 6+. I don't think that exists anywhere, but that is the toxic form. The 3+ form is the type that's normally there. And it can be linked to lung cancer if you take the toxic form. Yeah, I'm sorry, you're selling the wrong one. So this is the cool thing. They talk about this. One of the things they recommend for losing weight, and I don't know if it works, one of the things they recommend for losing weight is taking chromium. Why? Helps you get blood glucose into your cells. Taking carnitine. Why? What does carnitine do? Transports something. Transports fat into your cells, right? Carnitine is important for fatty acid oxidation and taking essential fatty acids, because essential fatty acids will preferentially be absorbed and your body will tend to ignore non-essential fatty acids, and then the essential fatty acids don't really metabolize for fuel so much as incorporated into your cells. So they recommend, if theoretically you want to lose weight, chromium, carnitine, and essential fatty acids. What form of chromium? 6 or 3? 3. Good. Fluoride functions, important in the hardness of bones and teeth. It promotes the synthesis of fluorapatite crystals. Fluorapatite crystals resists acid and dental caries. So fluoride is important for the health of teeth. Resists, what are caries? Anyone know dental caries? Cavities. Dental caries are cavities. So fluoride is important for dental cavities, which is why we do what with our water? Fluoridate it. Fluoride deficiency diseases, do you think we get these? Yeah, I guess if you're on well water and you never drink anything. Fluoride deficiency diseases causes dental fluorosis or dental caries. So you can get cavities if you don't have fluoride, or you can get the lack of fluoride in your teeth if you don't have fluoride. 3 to 4 milligrams a day, which I think we get more than enough of in water. No toxicity. Surprisingly. I'm sure there is. Yeah. I think eating toothpaste has other effects, not just fluoridation. There,
It's the fluoride. Like, my mom never let me have fluoride washes versus a tip. So why? Like, what's with the fluoride?
I don't know what the toxicity is. I looked it up at one point, but I don't remember now. I think I looked it up when I went through this class, because it's a big deal nowadays, because everyone drinks fluoridated water and it's everywhere and you're showering in it and, you know, yada, yada, yada. It's building up in different animals. So, all right. Well, yeah, I've heard that for many reasons, but I've been warned not to eat a tube of toothpaste for many reasons. Yeah, they're not supposed to ingest it. And fluoride is a problem. There is a toxicity of fluoride. He just doesn't need you to memorize it for Biochem 2. I'm not arguing with the fact that there's a toxicity of fluoride. I wish they didn't fluoridate our water. Yeah, same here. Okay, last 2. Cobalt. What's cobalt important in? Cobalamin. Fantastic. Cobalt's important for cobalamin. What the hell is cobalamin? Vitamin B12. What's important for the absorption of vitamin B12? Yep. What's important for the absorption of vitamin B12? I heard it, I thought. Intrinsic factor. Where does intrinsic factor come from? Stomach cells. Fantastic. Starts with a P. Parietal cells. Intrinsic factor from parietal cells will help through the absorption of vitamin B12. What's important for vitamin B12? Cobalt. Because cobalt is incorporated in the porphyrin ring, which is very similar to the heme ring. It's just instead of incorporating an iron, they incorporated cobalt, and that makes cobalamin. So it's a constituent of the vitamin B12. It's absorbed by the small intestine. Cobalt absorption is increased in patients with iron overload. Don't worry about it. That's a big TQ. It's important for B12, TQ. It's excreted in the urine, has low toxicity. Cobalt deficiency diseases cause vitamin B12 deficiency diseases. What do you think he's going to ask you about cobalt? If it's involved with vitamin B12. Low cobalt, low vitamin B12. Bam. Definitely know that. And lastly, the last 3 sentences of Biochem 2. Molybdenum. I don't even know what food's it in. It's a cofactor of metalloenzymes. Random stuff. Probably not going to ask you a single question about molybdenum. Zinc is the same. Zinc and molybdenum, metalloenzymes. And that's it. That's Biochem 2 in a nutshell. Dude, no, I refuse. Yeah. All right, so that's the end of Biochem 2. I'm sure you guys don't have any questions given that your test is coming up. So what do you need to know? Or do you want some random questions? You do have my mock exam online. I suggest taking my mock exam. I think it's very good. Zinc. Zinc and molybdenum are metalloenzyme cofactors. Just not advanced. That's the one part of my, well, there are many parts of my wardrobe I need to update, but my shoes are ridiculous. I've had these shoes for so many years. And they're the only shoes I can wear with pants with like wide bottoms to them. And I hate tapered jeans.
Me too.
I can't wear them.
I'm into flared jeans. Like, I'm old school.
I'm not going to say flared,
No,
but
I
I like
flared
big,
jeans. Like, that's what
yeah, bootcut jeans.
I did not like the most.
That's what this is, bootcut.
Those look a little flared to me. I did over your shoes.
That's the point. It is supposed to go over a boot, which is why it's a bootcut jean. All right. So you guys taking off, or are you sticking around for random questions?
Sticking around for random questions. We're taking off.
Cool. It's been a pleasure working with. Mainly being insulted by, laughed at, and called bald and garlicky. No, it's been nice. And you guys will see me around. It's not like our relationship is over. So, yeah, and you'll be asking me questions every try anyways, I know. And I'll be asking you questions. So what does this go through? Chapter 9? 9 through 12? 9 through 13? 13 is macrominerals. Okay. Cool. What's the common intermediate to all metabolic pathways? Acetyl-CoA. If you know it, say it loud and proud. I don't want to ask you what every time. You lazy bastards.
Lazy bastard.
Sorry, tutoring gods. Should probably turn off the mic. What amino acid is important for heme, glycine, and purine synthesis? I might have said something. I was thinking, what amino acid is important for heme, creatine, and purine synthesis? Glycine.
I like when you give us the answer, Paul.
Yeah, I know. It helps you a lot.
Merry Christmas.
Merry Christmas. How many pyrrole rings make a heme?
4.
Where is bilirubin conjugated?
Spleen.
Got to pick an answer. I'm not going to...
Spleen.
Conjugated in the liver. Bilirubin is conjugated in the liver. Yes. What does conjugation do to bilirubin? Yep, page like 102, 103. You should know what conjugation is. It's like conjugal visits. You're not going to forget this. So what happens at a conjugal visit? Sexy time. Fluid goes everywhere. You become more water soluble.
Nice.
Conjugation is making something more water soluble. I really got to show the vodka stuff. No, the people at home are going to be like, what the hell is he talking about? That was on page 103. Where do we have creatine?
Creatine muscle.
Muscle
And brain and weight.
and brain.
Weight, yeah.
Creatine is present in muscle, brain, and blood. Muscle, brain, and blood. What's the first nucleotide in purine synthesis? The first nucleotide in purine synthesis.
Is it M from the M? Or is it A?
I'll give you a hint. There are UMP and IMP. UMP or IMP?
UMP. Purines are UMP. Pyrimidines are IMP.
IMP. The first nucleotide in purine synthesis is IMP. Why can't it be UMP? What's U? Uracil. Is uracil a purine or a pyrimidine? It's a pyrimidine. So what's UMP, the first nucleotide in? Pyrimidine biosynthesis. So since UMA is a pyrimidine, it's got to be the first nucleotide in pyrimidine synthesis. Or because it's one of the first nucleotides, it's got to be involved in pyrimidine synthesis. The other one, IMP, is involved in purine synthesis. This is way above the level of where you guys are studying at.
No, it's so
2 days before the test.
Now I forget what purines and which ones they are. I'm thinking sulfide and something else.
What is an iron-requiring enzyme in purine and pyrimidine biosynthesis? Converts the reduced versions, or I'm sorry, the oxidized versions to the deoxidized versions of nucleotide bases. It only works on the D versions. The iron-requiring enzyme involved in purine and pyrimidine biosynthesis, specifically for making DNA, is ribonucleotide reductase. Iron-requiring enzyme takes the D versions, the diphosphate versions. Remember, not the tri versions, not the mono versions, and it takes them from the regular to the deoxy. So it takes TDP, thiamine diphosphate, to deoxythiamine diphosphate for DNA. So remember, it's important for DNA synthesis. What vitamin is important for DNA synthesis? Specifically, synthesis of the T bases. What? No.
It's that one that's
What's thymidylate synthase? It makes thymidylate, which is the T version. What vitamin is important for thymidylate synthase? Folate. Folic acid is important for thymidylate synthase, which makes the thymidylate base, makes the T base of DNA. This is a big test. What's the precursor of your catecholamines? Okay, good. Phenylalanine, which leads to tyrosine. He's probably going to ask you tyrosine. But phenylalanine is the true precursor because we use what vitamin to do? What does phenylalanine turn into? Turns into tyrosine by ring hydroxylation. What vitamin ring hydroxylates? It's not one of the vitamin chapters from chapter 8. Tetrahydrobiopterin, BH4. What's the precursor of melatonin? See you around, buddy.
Goodbye.
Text me or call me if you can't get that test, and I'll email it to you. Tryptophan, precursor of melatonin. And serotonin. And what else? Tryptophan does melatonin, serotonin, and what vitamin is tryptophan the precursor of? NAD. Tryptophan is the precursor of the nicotinamide portion of NAD. Or NAD is not a vitamin. What vitamin is NAD? Vitamin B3, niacin. Good. What amino acid precursor is required for thyroid hormones? Tyrosine. What amino acid precursor is required for a thyroid hormone? Tyrosine. What class of chemical reaction results in the single-step production of an inhibitory neurotransmitter? Before you blurt out answers, listen to it again. What class of reaction results in the single-step formation of an inhibitory neurotransmitter? Decarboxylation. What am I talking about? The synthesis of GABA. The synthesis of GABA is the one-step process of making an inhibitory neurotransmitter. Like I said, that's been on a few tests. That TQ will probably show up. And the one step is decarboxylation, because glutamic acid or glutamate decarboxylase does that. What is the precursor to endothelial? What's the precursor to nitric oxide? EDNO is endothelial-derived nitric oxide. What's the precursor to it? They're the same thing. The endothelial-derived relaxing factor is endothelial-derived nitric oxide. But what's the precursor to it? Amino acid. You guys are really weak on this amino acid chapter. Arginine. Arginine is the precursor. That's chapter 9. That's all chapter 9. Brush up on chapter 9. That's the last chapter, like the first chapter we covered for this test, and that'll come up. He emphasized the what? No, I think you get a pretty equal spread, but you're going to get chapter 9 questions. You have four ones on this test. I'm betting not a lot's going to come from chapter 10. Chapter 10 has one TQ, acetyl-CoA is the common intermediate of the metabolic pathways, and he's going to talk a little bit about like in what state do you have muscle breakdown to provide energy to the liver, starvation state. In what state do you have fat breakdown to provide energy? In the fasting state. In what state are you, building glycogen stores in muscle? In the fed state. So stuff like that. What's the definition of fasting? Because he's not going to say fed, fasting, whatever. If he asks a question, he's going to say, this man recently ate. Or this man ate within 12 hours. Or this man ate like 12 hours or 3 days ago. What? Nouns.
Past 12 hours.
I've taken some nouns out to imitate the man ate. Man eating 2 hours ago. I really shouldn't do this stuff on vodcast. You guys are a bad influence. Bad. Hey. I know. What tissue utilizes the most glucose in the body? The brain. What does postprandial mean? Postprandial means after eating. Prandial, make sure you know that term. What's the normal amount of time for the postprandial level of glucose to return to normal? 2 to 3 hours. Good. Oh, yeah. True or false? Vitamins supply energy from their breakdown. False. No. True or false? Vitamins are required in very large amounts in the diet. True or false? Vitamins are not utilized for structural purposes. True or false? Vitamins are not utilized for structural purposes.
They are utilized, right? Is there cofactor or they're not?
Structural.
Oh, structural.
False. True. Vitamins are not utilized for structural purposes. True.
Okay, they're not. Okay, bump.
What vitamin is a substrate? Actually, what vitamin is a substrate?
What vitamin is
Yep.
No.
I think you said vitamin. Okay, I'm going to say it a little more. What vitamin is a substrate for ADP ribosylation reactions? NAD. It's the only vitamin that's ever a substrate for anything. ADP ribosylation, yeah.
Say that one more time. So the only answer to that would be NAD?
Yep, NAD is the only one that's a substrate for anything. It's a substrate for ADP ribosylation reactions. What fat-soluble vitamins can be stored? A, D, E, and K. What water-soluble vitamins can be stored? C and B12.
What about D?
D is fat-soluble. All the fat-solubles can be stored.
The only water-soluble ones are B, C, and...
All the B's, C's, folic acid, biotin. The water-solubles? B12, vitamin C, and your fat-solubles. Yeah. The rest are just freely excreted. Okay? What gives you scurvy? Vitamin C deficiency. Good. Ooh, what gives you megaloblastic anemia?
Ooh, 9 and 12.
Nice. B9, B12, which are known as?
Macro something.
What are B9 and B12? What are the names of them? Cobalamin and that's B6. Folate. B9 is folate. You don't have to know that for Sarkar. So when I say B9, B12, make sure you know that he's going to say B12 and folate or cobalamin and folate. B9 is folate, but he doesn't refer to it as that. And I asked my nutrition professor, like, why does no one refer to folate as B9 and biotin as B7? He's like, I don't know. He's like, they are B9 and B7, but nobody says that. I'm like, it's easier.
Yeah.
He's like, maybe because B9 sounds like benign, which could be mistaken for something else. Okay. What causes microcytic anemia?
Ooh, that's
B6, aka what? B5. Pyridoxal phosphates, B6. Or what? B6 or?
no, it's one of the
It's one of the minerals.
yeah.
Iron. Iron causes what type of anemia? Iron causes what type of anemia aside from iron deficient anemia? Smartass.
The micro one.
Hypochromic, microcytic. Good. Or microcytic hypochromic. Either way, it's both. What causes methmalonic aciduria or methmalonic acid in the blood? Be what? Nice job. Because methmalonyl breaks down, B12 helps in the breakdown of methmalonic acid. So if you don't have B12, you get methmalonic acid in your blood, in your urine, etc. Nice job, man. What's pernicious anemia? Not a B12. Well, yes, that's true. It's a B12 deficiency, but it's a very specific type of B12 deficiency. What causes the B12 deficiency that allows it to be pernicious? It's a deficiency of intrinsic factor leading to a deficiency of B12. So pernicious anemia specifically means you have low B12, low intrinsic factor causing low B12. Causing what type of anemia? Megaloblastic, microcytic. Which one? Megaloblastic, B12. So, intrinsic factors secreted by the parietal cells of the stomach. Cobalamin, B12 flows through the stomach. Down in the small intestines, intrinsic factor and vitamin B12 bind. And the small intestine receptors to absorb vitamin B12 will only recognize vitamin B12 bound to intrinsic factor. The receptor's substrate is intrinsic factor bound to B12. They must both be there or they will not be absorbed. You will not get your intrinsic factor back and you will not get your B12. So you must have them both. When they are both present, it is absorbed. So that's it. It allows for the absorption of vitamin B12.
That's its own purpose.
Mm-hmm. Now, this is where he gets more complex than that. So once you absorb it, it's turned into hydroxycobalamin, right? And it's transported in what? How is B12 transported? Transcobalamin 2, because 2 does transport. And then once it gets to the tissues it cares about, this is on page number B12, whatever. Yeah, this is the novel. Page number, son of a gun, 142. So, cobalamin is absorbed by the intrinsic factor cobalamin receptors. It's turned into hydroxycobalamin. It's transported attached to transcobalamin 2, 2 for transport, T and T go together. When it gets to the tissues, it's dumped out as hydroxycobalamin and it has 3 fates. It can be turned into, or it can be attached to transcobalamin 1. Transcobalamin 1 stores it. Or it can be turned into, where were we? Deoxyadenosylcobalamin. If it goes into the mitochondria, it can be turned into deoxyadenosylcobalamin, where it catalyzes the conversion of methmalonic acid to succinyl-CoA. The reason you know that one's in the mitochondria is because that's the only place you need succinyl-CoA. That's where the TCA cycle occurs. So the formation of methylmalonic acid to succinyl-CoA by deoxyadenosylcobalamin happens in the mitochondria. In the cytoplasm, it's turned into methylcobalamin, where it does two things. It does methyl tetrahydrofolate to tetrahydrofolate and homocysteine to methionine in the cytosol. So what it does is it takes the remember B12 is a methyl transfer. It takes the methyl group from methyl tetrahydrofolate, making it tetrahydrofolate, and it gives it to homocysteine, making it methionine. Coolish? What food contains thiaminase? Raw fish. What does thiaminase in excess do? Deficiency of? Thiamine. Good. What kind of diet causes the deficiency of thiamine? High polished rices and stuff like that. That was the worksheet you did? Yep. What two types of reactions is thiamine involved in? Oxidative decarboxylation and transketolase or transaldolase, transketolase. So who does decarboxylation? It's B6, good. B1 does transketolation and oxidative decarboxylation. Oh yeah. Vitamin B6 does decarboxylation. Vitamin B6 also does transaminase or aminotransferase transaminase. Have a good night, guys. Thanks for the gummy bears. I haven't eaten them yet. I told my girlfriend to pick up a bottle of vodka to soak them in. Dang it, vodcast. Now they know I get paid in gummy bears and I soak them in vodka. Once they're soaked, I'll bring them back. Yeah. They're amazing. You'll try them now, won't you? Yeah. We've been, it's terrible because I hate the maraschino cherries. There's a place we used to go to on the river that my uncle lives down on, and they have just bottles of maraschino cherries. They pour the juice out and fill it with Everclear, and you pay like it's 4 cherries for $1, and $1 will get you going. Yeah, moonshine. Oh man. All right, so thiamine does oxidative decarboxylation and transketolation. What's the disease of deficiency in thiamine? Beriberi. You guys better know thiamine well. What kind of things cause water-soluble vitamin deficiencies in general? Alcoholics. Eating. Processing. Light. Oxygen. Diuretics. Dialysis. Cool? Just in general. What's the AKA for vitamin B2? Vitamin B2. Riboflavin. What's the disease associated with riboflavin? It's the easy one. A. Riboflavinosis. Without riboflavin. A. Riboflavinosis. What's the active form? What's the active form of thiamine? TPP. Good. TPP. What's the active form of riboflavin? FAD or FMN. What types of reactions is riboflavin involved in? Oxidation reduction, oxidoreductases, because that's what FADs and FMNs do, is they oxidize and reduce stuff. What's the AKA for vitamin B5? B5. I skipped because of that. Pantothenic acid. Pantothenic acid. What's the active form of B5? Pantothenic acid. It's CoA. Just CoA. Or acyl carrier protein, ACP. Where did you see ACP? Nice job. Fat bison. Oh, okay. That's right. I was like, wow. The active form that you really care about is coenzyme A. What's coenzyme A involved in? Everything. Coenzyme A. So what's the common intermediate in all metabolic pathways? Acetyl-CoA. What vitamin is important for acetyl-CoA? Coenzyme A. What vitamins make acetyl-CoA? B5 is important, yes. Do you guys know what I'm talking about? What enzyme makes acetyl-CoA? Pyruvate dehydrogenase complex. TPP? Wow. FAD, NAD, lipoate, and CoAsh. So you need B1, B2, B3, and B5 to make acetyl-CoA. Yeah, the first 4 water-soluble vitamins. Okay? What's the active form of niacin? NAD, NADP. What type of reactions is it involved in? Oxidoreductase. And what's it a substrate for? ADP ribosylation is NAD. Good. That would, yes. So, but what are dehydrogenase enzymes? Oxidation reduction enzymes. Because what does a dehydrogenase do? It takes hydrogens, it moves electrons. So dehydrogenase, oxidase, and reductase all are oxidation reduction enzymes. So if you see a dehydrogenase, an oxidase, or reductase, you're thinking B2 or B3. Yeah, riboflavin or niacin. What vitamin is niacin? B3. Good. Ooh, what's the disease associated with niacin? Sugar. It's pellagra. The disease associated with niacin is pellagra. And what amino acid is important for the creation of niacin? Tryptophan. Tryptophan makes niacin, makes the nicotinamide portion of niacin. So what are the signs and symptoms of pellagra? 5 Ds and a W. Digestive, dermatitis, depression, death, and weight loss. Oh, and dementia. I missed one. Depression, digestive, dermatitis, dementia, death, and weight loss. Not necessarily in that order. Okay. What's vitamin B4?
There's no such thing.
Good job. I'm sure there actually probably is, but not that we need to know, because I don't know it either. What's vitamin B6? Pyridoxal phosphate. What reactions is it involved in? Transamination, aminotransferase, or decarboxylation. So give me an example of a transamination. Pyruvate alanine, 1 down. Give me another one.
Oh my gosh.
There's 3 you gotta know. Alpha-ketoglutarate 2?
Glutamate.
Glutamate, nice job.
Yes. Pull that one out my ass.
And oxaloacetate 2? Aspartate, nice. Those will be your example questions for that one. What's your example question for thiamine?
Thiamine B1. What about it?
That would be the transketylation, but that's less likely to be asked. What's the key example question for thiamine? We talked about it a little bit.
Thiamine's the only something, right?
Pyruvate to acetyl-CoA.
Oh.
Thiamine, TPP, pyruvate to acetyl-CoA. There's also alpha-ketoglutarate to succinyl-CoA. Why?
Because it goes in the TPP cycle.
Because pyruvate dehydrogenase complex and alpha-ketoglutarate dehydrogenase complex both need the same 5 cofactors. That's good old Biochem 1. Believe it or not, guys, it took me like 4 tutorings to get this stuff down. Like, you're like, oh my God, like he remembers all this stuff. I've been through it 4 to 6 times, so don't worry about it.
I mean, like repetitive. Like, you mean 4 to 6 times as opposed to 4 to 6 sessions, or like 4 to 6 times, like?
I've taught this 4 to 6 times.
Thank you, amen. Okay, I was a little worried.
No, I've taught this. Like, the first time through was not this smooth. No, that's fine. Yeah, because I took it a second time. All the tutorings, I took a second time with Ian so I could get ready to tutor them. And then I taught them a few times.
That's a good idea for boards, though.
It is. It's way more in-depth than you need boards.
Okay.
I mean, I had a smooth time with Biochem on boards, I'm not going to lie. But I put a lot of time in it. A lot of my friends did just fine, and they didn't put near as much time into Biochem as I have. Yeah, I took board reviews. They were worth it. I think a lot of people can pass without board reviews, but I think they were helpful. All right. I'm not a genius. Thank you. I'm a balding garlic guy. Garlicky balding guy.
And it's practically genius, right?
Maybe that's why all my first dates go so bad.
Oh.
You guys take me way too seriously. All right. We talked about that. Ooh, vitamin B6 deficiency causes what type of anemia? Microcytic anemia. Big one you got to know, microcytic anemia. Hard to pull out of your... Yeah. Causes dermatitis and mental confusion and convulsions. What was the other one that caused microcytic anemia? B6 and iron. Second time in a row. Nice job, man. B6 and iron. That's the most frustrating part. Oh, yeah.
Is this 9 through 13? Is that what you're saying?
No, it's 9 through 12.
Or 9 through 12, okay.
Yeah. What's the AKA for vitamin B12? Cyanocobalamin. What does it need? It needs a cobalt. What does it need to be absorbed? Intrinsic factor from the parietal cells. What are the active forms of cobalamin? Methylcobalamin, present in the cytosol or the mitochondria? Methylcobalamin is present in the cytosol. What's the active form in the mitochondria? The Oxidized So, vitamin B12 is bound to intrinsic factor where it's absorbed by the intestinal mucosa and then it's attached to blank to be transported to the tissues.
You just said the cobalt transport.
What? Yeah, what transports cobalt? Transcobalamin.
Yeah, there we go.
Transports. Transport is 2. Transcobalamin 2 transports. TNT. It gets to the cells. It's turned into hydroxycobalamin. It's then bound to transcobalamin 1 for storage. Or it's dumped into the mitochondria to be turned into deoxyadenosylcobalamin where it catalyzes methmalonyl-CoA to succinyl-CoA. Or it's dumped into the cytosol to be turned into methylcobalamin where it does homocysteine to methionine. And methyl THF to THF. So what are two major diseases of B12 deficiency? Homocystinuria, bam, and pernicious anemia, and the third one, methmalonic aciduria. You can't convert methmalonic acid down, you can't convert homocysteine down. You get a buildup of methmalonic acid and a buildup of homocysteine. And believe it or not, the best test for B12 deficiency is a level of methmalonic acid. Okay. Why is B12 so important? Why do we care? I mean, aside from pernicious anemia and methmalonic aciduria and homocystinuria, what system in your body is it essential for? Nervous system. It causes demyelination, which is one of the big reasons that if you see something like megaloblastic anemia, which is caused by what?
B12 and B9.
B9, B12, and what's B9? It's folate. Benign, don't worry about it. If you see megaloblastic anemia and you just give them folate, the megaloblastic anemia could go away, but they're still, if it was caused by B12, they're still going to have a B12 deficiency. They're still going to be demyelinating their peripheral nervous system, but you're masking the anemia. So they're still losing nervous tissue and getting peripheral neuropathies, but you're masking the anemia. So be careful what you supplement. Make sure it's the right thing for a patient. Folic acid is derived from a polyglutamate conjugate with how many glutamate residues? Poly actually is apt in this point, but you need to know it has 7 glutamate residues. That was a TQ I told you guys. 7 glutamate residues from the polyglutamate conjugate, where it's cut down to 1 by the enzyme gamma-glutamyl hydrolase. Maybe on there. What enzyme makes the active form of folate? Well, what is the active form of folate? Tetrahydrofolate. Tetrahydrofolate. What enzyme makes the active form? Dihydrofolate reductase. Why reductase? Because it's adding H's, and adding H's is reducing. So for God's sake, on the test, if you know that folate is the inactive form and tetrahydrofolate is the active form, it has 4 H's, pick the enzyme that says, I reduce you. Does it twice. And what vitamin and coenzyme does it need to do that? NADP, which is vitamin B, vitamin B3, because all biosynthetic reductive processes involve NADPH. All biosynthetic reductive processes involve NADPH. NADPH is vitamin B3. It's involved in the reduction of folate to tetrahydrofolate, which you know because you're adding H's. What important, that may be too vague. We talked about these already. The 2 reactions that B9 is involved in. Serine to glycine is a good one. Serine to glycine is an example reaction. DUMP to DTMP. What's that called? Thymidylate synthase, good. Thymidylate synthase takes DUMP to DTMP, that's a big one for folate. And folate's also involved in homocysteine to methionine because it gives its methyl group to B12. What's the active form of B12 that picks up the methyl group from folate? The active form of B12 that picks up it becomes methylcobalamin. So cobalamin becomes methylcobalamin by taking a methyl from methyl tetrahydrofolate. So tetrahydrofolate donates a methyl to cobalamin, making it methylcobalamin. Methylcobalamin donates it to homocysteine to make methionine. Does that make sense? So these guys all kind of interweave together. Yeah, they're very sweet. What type of anemia does folate deficiency cause? Megaloblastic, aka macrocytic, but megaloblastic is what he's going to ask. What is B9 involved with that's so bad? Cancer would be one. DNA synthesis. So where do you see defects? Central nervous system, neural tube. Folate is one of those massive prenatal vitamins that they push in all women because folate is important for neurological development. And it causes cancer because of deficiency of thymidylate synthesis, which causes DNA problems, which can cause cancer. What about biotin?
What about it? B9. No, that's folate.
You don't need to know. It's B7, but nobody cares. What's the active form of biotin? That's vitamin B6. Carboxybiotin. Ooh, this one's going to be good. What type of reactions does biotin participate in? Carboxylation. Does that make sense? The active form is carboxybiotin because it carboxylates. Transfer a carboxyl group, but yeah, a carbon. And big TQ, it's made by intestinal bacteria. Biotin is made by intestinal bacteria. So anything you see a carboxylation in. What food group contains avidin that breaks down biotin? Biotin. Egg whites. What do you have to do with your egg whites to make sure that you don't break down biotin? You have to cook them. Good. Avidin. Avidin, like thiaminase. Not as easy. What's the big deficiency condition of biotin that he's going to ask you about on the test? Hair loss. AKA, what's hair loss called? Alopecia. Good. What's the active form of vitamin C? Ascorbic acid. What is the enzyme that makes the active form of vitamin C? But gluonolactone oxidase. Nice job. Ironically, I think this was a question on our last pathology test. The only 3 animals on Earth that cannot make their own vitamin C from glucose, because vitamin C comes from glucose, it's like humans, hamsters, and guinea pigs or something like that. It was a random question on our test, but I was like, holy crap. Like, humans, hamsters, and guinea pigs cannot make vitamin C from whatever. And that's in pathology. See, this stuff comes back. That was farm talk, sorry. Gluonolactone oxidase. Gluonolactone oxidase basically takes glucose and turns it into vitamin C. Gluonolactone is because from glucose you go to gluono, and then gluonolactone and all that stuff. So just GLU, glucose, is a precursor to ascorbic acid. Ooh. Is vitamin C stable to freeze? Yes. Vitamin C is okay with being frozen. Is it heat labile? What's labile mean? Labile. It's destroyed by. Is vitamin C heat labile? Yes. It is the most heat labile. It is the most sensitive. It is the least stable TQ of all water-soluble vitamins. What's the RDA of vitamin C? 100 milligrams a day. Make sure you know the units because he might mess with you on units. It's unlikely, but he might.
No, he would.
And that is 100. He would. That's correct. I mean, it's just average content.
Yes. Yeah. What are the main things vitamin C is involved in? It's an antioxidant. Nice job. What type of enzyme? Monoxygenase and dioxygenase. It adds oxygens. And the 2 you got to know, it makes norepinephrine biosynthesis and collagen synthesis. Vitamin C collagen synthesis. Why? How do you know that? Because scurvy has what symptoms? Bleeding gums, which is because you have a lack of collagen. So scurvy is from a lack of vitamin C, and the symptoms you see are from a lack of collagen. So no collagen, you can't hold wounds closed. You can't hold your skin closed. You get bleeding gums. So vitamin C is collagen hydroxylation, norepi biosynthesis, and monooxygenase and dioxygenase. It adds oxygens to stuff. It is also a very important antioxidant. Norepinephrine biosynthesis. It is also important for iron absorption, and that's on page 152. It increases the absorption of iron in the small intestine because it keeps iron in its reduced form, which is ferrous iron. The big part about vitamin C is that it regenerates vitamin E. Vitamin E is your primary antioxidant, and the way it keeps antioxidizing stuff is because vitamin C comes and antioxidizes vitamin E after vitamin E has antioxidized something. So once vitamin E reduces something, it oxidizes itself, and then vitamin C comes along and antioxidizes vitamin E, and then vitamin E goes back out and does its job. It's like the IV after a long night of drinking, so you can keep drinking. Good analogy? After your Everclear gummy bear. Okay, vitamin C causes scurvy. Vitamin C deficiency causes scurvy. All right, so let's float through a few fat-soluble questions, and then you guys are out of here. What are all fat-soluble vitamins a derivative of? All fat-soluble vitamins. Are you reading out of the book? Do not answer questions if you're reading out of the book. That is a cardinal rule. You can think them to yourself, but do not answer. All fat-soluble vitamins are derivative of isoprene units. What are isoprene units built of? They're involved in cholesterol synthesis. They're built of acetyl-CoA. Because remember, in cholesterol synthesis, we took 3 acetyl-CoAs, slapped them together into an isoprene. So we're making the same thing. Isoprene units are the beginnings of cholesterol, the beginnings of fat synthesis. So these guys are fat-soluble vitamins. They're steroid-like vitamins, not all steroid-like. So are they hydrophilic or hydrophobic? Hydrophobic. Are they polar or non-polar? Non-polar. What can lead to malabsorption of fat-soluble vitamins?
Not enough fat. Triacylglycerol. Something like that?
Chylomicrons, sure, if you have lack of chylomicrons. But what's the big thing? What happens if you have cholecystitis or gallbladder dysfunction? If you can't digest fat, you can't get fat-soluble vitamins. So if you have no bile, if you have gallstones, if you have a gallbladder removal, you can't get fat-soluble vitamins, because to absorb something that's fat-soluble, you need that bile which creates the micelles, right? Okay. What's vitamin A involved in? Vision. Vitamin A deficiency is the leading cause of blindness in the world. TQ, vitamin A deficiency is leading cause of blindness in the world. What are the active form of vitamin A? Retinol, retinal, and retinoic acid. Retinaldehyde, retinalcohol, and retinoic acid. What is the most active one? Retinol, OL, is the most active form of vitamin A. What's the plant version of vitamin A? Beta-carotene. What's the animal storage of vitamin A? Retinol esters. Remember, like cholesterol esters were the storage form of cholesterols? Retinol esters are the storage form of vitamin A. Okay, here's the fun part. So, don't really care about that. That might be important, but I'm going to skim by it. So, which form is important for vision? Retinal. Al is important for vision because it forms 11-cis-retinal, which combines with opsin to form rhodopsin, which is present in what cells? The rods of your eye, which when it's exposed to light breaks back into opsin and all-trans retinol. So, in the dark, what are your levels of GMP, cyclic GMP? In the dark, what are your levels of cyclic GMP? They are high. In the dark, your levels of cyclic GMP are high. And in the light, what are they? Low. Why? What is the G protein that is activated by light that breaks cyclic GMP? The G protein that's involved in the light detection pathway is transducin. So in the presence of light, transducin is a G protein, but transducin doesn't make cyclic GMP like adenylate cyclase made cyclic AMP. Transducin turns on cyclic GMP phosphodiesterase. Transducin turns on cyclic GMP phosphodiesterase. What does cGMP phosphodiesterase do? Breaks down cGMP. So just like CAMP phosphodiesterase broke down CAMP, cyclic GMP phosphodiesterase breaks down cyclic GMP. So when the light hits transducin, or when the stimulus gets to transducin, whatever it is, it turns on cyclic GMP phosphodiesterase. Cyclic GMP phosphodiesterase decreases cyclic GMP levels. So in the light, you have low cyclic GMP levels. In the dark, you have high cyclic GMP levels. Now here's the one other key thing. He's going to ask you about the cyclic GMP levels. He's going to talk to you about transducin. And mainly that transducin is the G protein that's involved. And he's going to talk to you about whether the cell is hyperpolarized or not. So, when would you want your rod to be hyperpolarized? What is hyperpolarized? Does that mean it's activated or it's inactivated? Hyperpolarized means it's inactivated. Depolarized means it's activated. Hyperpolarized is when it goes way down below the curve. So when would you want your rod to be hyperpolarized? When would you want your rod not to fire? What do rods pick up? Light. So when it's too bright, you want to turn your rods off, because otherwise it's just going to white out, right? So in the light, you hyperpolarize the cell because you don't want the rods firing. You want the rods to shut down, or all you're going to perceive is light. In the dark, the rods come back up to normal so they can depolarize and pick up the little light stimuli. But if you walk outside in the bright sunlight, what's it like at first? You can't even take it. You can't keep your eyes open because it hurts. It burns. There's too much light until your rods hyperpolarize and shut off. So the key is you want your rods hyperpolarized. Hyperpolarized means they're off, and you want them off in the light. Make sense? So he's going to talk to you about whether they're hyperpolarized. They hyperpolarize in the light. He's going to talk to you about the cyclic GMP levels. The cyclic GMP levels are what in the light?